This video will cover topics related to neuronal membrane potential and a brief introduction to the synaptic transmission of neurons. To begin, let's go over membrane voltage. Voltage is defined as the difference in electrical charge between two locations, usually measured with a voltage meter containing both a positive and a negative probe wire. In neuroscience, the convention is to place the positive probe inside the neuron and the negative probe outside the neural membrane in the extracellular fluid. We call this the transmembrane voltage of a neuron, and it's measured using an invasive technique called intracellular patch clamp recordings. The resting potential of a neuron is the transmembrane voltage at rest, and it is calculated to be approximately negative 70 millivolts. This means that the inside of the neuron is more negative than the surrounding extracellular fluid. This is due to the concentration of ions in and around the neuron. The movement of these ions across the membrane is responsible for nearly all of the neural activity we observe. For that reason, it's important to consider the relative ion concentrations inside and outside of the cell. In a resting neuron, there are more potassium ions inside of the cell, and there are more sodium, calcium, and chlorine ions outside the cell. Remember that the transmembrane voltage of a neuron at rest is about negative 70 millivolts, which seems odd since there are more positive ions concentrated outside of the cell. Shouldn't we expect the transmembrane voltage to be positive? Well, not necessarily. Although neurons are driven by an electrical force, based on the charge of the inside of the cell, they're also driven by a chemical force, which has to do with the concentrations of the specific ions inside and outside of the cell. Equilibrium doesn't mean the ion wants to be concentrated inside and outside of the cell equally. In fact, each ion has its own preference for how it wants to be distributed across the membrane. This is called the ion's equilibrium potential, and we'll talk more about it in a moment. But first, let's look at the structure of the neuronal membrane. The membrane is a fluid mosaic of complex protein structures which acts as a selectively permeable ion channel. Potassium channels are always open, meaning potassium ions are free to flow in and out of the cell. Sodium channels are closed at rest, so they cannot flow freely across the membrane. To determine the transmembrane potential of a neuron, we use the Nernst equation, which is fairly complicated. Put simply, we need to know the temperature in degrees Kelvin, both the concentration and valence of the ion, and then plug all that in with Faraday's constant and the gas constant to find the transmembrane voltage produced by a specific ion. We do this over and over until we considered each of the ion's Nernst potentials. Because each ion is driven by both a chemical and an electrical force, we need to express the total force acting on an ion at a given point. The total force is called the driving force, and it is equal to the sum of the chemical and electrical force. When the driving force on an ion is zero, meaning the ion is no longer moving across the membrane, the ion is said to be at electrochemical equilibrium. As a side note, all ions attempt to reach their equilibrium potential, thereby affecting other ions' ability to reach their own equilibrium potential and affecting the membrane potential altogether. This changes the driving force of all the ions, creating a constant tug of war between the ions. Here are a few types of gated ion channels you'll see in your behavioral neuroscience course. There are ligand-gated channels in which a chemical called a ligand binds to these channels to open or close them. Voltage-gated channels that are triggered, meaning opened or closed, when the membrane voltage reaches a specific target range. Mechanically gated channels, which are found especially in the peripheral nervous system, they open or close by a mechanical force such as touch. And then there are optically gated channels, which are opened or closed by light. To communicate the activity of neurons, we have to be careful with the language we use, and so there are a few more terms we need to go over. Hyperpolarization is when the membrane potential is made more negative, making the neuron less likely to fire. Depolarization is making the potential more positive, thereby making the neuron more likely to fire. Inhibitory postsynaptic potential, or IPSP, is a hyperpolarization of the neuron, and an excitatory postsynaptic potential, or the EPSP, is a depolarization of a neuron. That peak that you saw in the graph corresponds to the rush of sodium ions into the neuron, causing the neuron to depolarize and fire an action potential. An action potential is defined as a depolarization of the neural membrane, resulting in a brief conduction of electrical stimulation. Again, this occurs when sodium channels open briefly, allowing the sodium ions to rush into the neuron and reach their equilibrium potential of positive 60 millivolts. Recall that sodium ions are more concentrated outside the cell at rest. The action potential can only be initiated once the neuron reaches its spike threshold of negative 55 millivolts. Once initiated, the neuron will fire. This is called the all or none law. Via synaptic integration, a neuron is able to sum the IPSPs and the EPSPs to modulate its transmembrane potential. However, not all ions are created equal. Some carry more positive charge than others. Additionally, modulatory synaptic inputs help tip the ionic scale, making some inputs more or less effective at depolarizing or hyperpolarizing the neuron. IPSPs and EPSPs must travel from the dendrites to the axon hillock where they are integrated. Here's how they travel. There's spatial summation, which is the summation of two presynaptic action potentials occurring simultaneously at two different parts of the neuron. This results in a bigger EPSP than the individual presynaptic potentials, thus making the neuron more likely to fire. Temporal summation is when a single presynaptic neuron generates many spikes in rapid succession onto the postsynaptic neuron. The neuron is a weak electrolyte, meaning it conducts electricity but not well enough to carry a signal from the dendrites to the integration zone on the axon hillock. As a result, the EPSP will become weaker and weaker as it travels across the neuron. By firing onto a neuron in rapid succession, the signal becomes impervious to its loss in potential as it makes its way to the axon hillock.